This media was gratuitously produced by Clear Contributions Audio and Video. The intellectual property contained within belongs to Dr. Richard Gardner and Garland Science. Clear Contributions. Let's have clarity.
All right, before I forget, after Thanksgiving break, after Thanksgiving break, on Monday, I'll probably send out a sign-up sheet for that week. So if people can read these articles and then get together with your people like really soon, because we have very little time here. So during that week and maybe Monday, and then we'll have to figure out presentation schedule. We have then 3, 6, 9, 12, 6 presentations. So we can actually do it we could probably do it the last 2 days of class instead of the last 3 days. Have an extra day for review or whatever. Or we could do one presentation on the third to last day of class and put everybody else. Again, if we have 3 presentations in a day, it's 12 minutes, because you can't, if you go overtime and it's 15 minutes, well, that might work. 15 is 45 minutes. People coming up and moving back. One thing we shouldn't have so much this time was had in the past is people set their computers and hook it up to this thing, and the projector in their computer doesn't match or whatever, technology issues. If you have stuff on your iPad, it should work on this screen. So that could save us some time with all of our technical glitches that we always have. I don't know why the Fox News is on here. There's some interesting stories, but I don't know why that ended up on there. What we're gonna do is, I know what I was doing. I was looking for our class website, not the email. Dang it. Class website. Where's our class website? Is this where it is? Yeah. Come on. All right, I had it somewhere. I thought I had it up somewhere. There it is, my SVU. All right, let me just make sure that. So the next homework due will be Wednesday after Thanksgiving, and then Friday. So it's two classes in a row, and they're on those other 3, and then 2, so 5 total other papers, and they are down here. So if you kind of scroll down. Weiner, Checkpoint paper, Hartwell, Nurse, Masui, and Hunt. And I'm not sure why. Oh, yeah, never mind. Not the original Checkpoint paper. It's these essays. So the one Hartwell essay, Nurse essay, Masui essay, Hunt essay, and Weiner essay. So all these ones say essay on them. So it's the same like these other ones we just read from Kirshner and Mitchison. So these are 5 people that came up with fantastic research into the cell cycle. And so for the next homework, which will be on Wednesday, November 29th, before class, the same thing. We're going to discuss 3 of those. If you look in the homework, and I think it's like probably those first 3 listed, Hartwell, Nurse, and Masui, and then Hunt and Weiner, but don't quote me on that. So look up those and then read those over break, as well as the other papers. I know this is a lot of stuff at the end of the semester, but these are all fun papers. So they could be relaxing for you. I mean, they're not hard to follow. You just kind of read them, and then there's. What I would do is there's some questions again on the homework about the paper. So I would read the questions first. So then as you read the paper, you have in your mind what it is that you're kind of looking for. But then read the whole paper anyway. You can do one every day, and then you'll get them all done. And so the last. Wednesday and Friday after Thanksgiving, we'll talk about those. So we start doing those, as well as this other paper. These other papers are going to be much more difficult, these research papers. Let's see, who else had the FlipAce papers? Travis, Blake, and Amy. I've got 2 FlipAce papers left. Who didn't get one?
I don't know if you guys don't mind, but we'll take our time. Thank you.
All right. we were talking about collagen the other day. Again, here, this one shows how the collagen, one protein is braided up, so you got sort of three of them, sort of a trimer, but they're kind of braided in a cool way. And then that's what gets secreted from the cell. So inside the cell, and then that's secreted outside the cell. And then you start having the assemblies into the larger and larger versions of collagen outside the cell, because it's by that time it's too big to fit out, to go through the cell. So again, by the time you get from the triple stranded, that can leave the cell, but the collagen fibril or the fiber, which is a bunch of fibrils put together, that's too big. So that's all kind of put together outside the cell. And then the fibroblasts are what, these are the cells that crawl around and leave the collagen in their wake. I just like this one. This is a collagen defect. And it's this guy that's some circus, pay 10 cents and watch him pull his mouth apart or whatever. All right, so that's sort of collagen. Integrins. So integrins are proteins across the cell membrane. They connect to the cytoskeleton on the inside. They connect to the extracellular matrix on the outside. So it's just sort of a connection. Here they are going through the cell membrane, connecting on one side, connecting on the other side. Not a whole bunch more to talk about integrins besides that. They can form a sort of an inactive integrin like this. It's kind of like transformers, and they can kind of unfold and then bind to whatever they're supposed to bind to on the outside, and then they cross the membrane. All right, so we've talked a bunch about different extracellular matrix proteins such as collagen, keratin, and integrin proteins. Then we also have extracellular matrix polysaccharides. And so, let's see if I can get this to come up correctly. This is a common extracellular matrix polysaccharide. It is the GAG, which stands for glycosaminoglycan. And that is the larger category, and this particular one is hyaluronan, and I can hardly say that word. But if you see the word hyaluronan, that is a particular GAG, and a GAG is basically glycose should remind you of sugar, and amino is like, notice how some of these sugars have ends on them, nitrogen atoms, that's sort of your amino group, so it's sort of an aminoglycan, so you've got, this particular one's a disaccharide. There's two different kinds of sugars. Don't memorize what they are, but just remember there's sort of a fancy polysaccharide made of a repeating disaccharide, and it's called a GAG. And from GAGs, then you can build up proteoglycans. We talked before about glycoproteins and proteoglycans. Glycoprotein is a protein with a bit of sugar on it, and proteoglycan is a sugar with a bit of protein on it. So it's color-coded here. The proteins are green and black, and the GAG is red and blue. So the blue thing here and the red stuff here is all sugars. Notice how much sugar there is here, and then you've got a few proteins attached. So the little black balls are proteins, and these green lines coming down are proteins. But it's more, again, the protein is, let's see if I can get it to work right. This protein here is sort of attached to the sugar. So it's mostly a sugar with proteins then attached to that, and then sugars attached to that. So that's sort of your proteoglycan. So the relationship of proteoglycans and GAGs, then to summarize, which one is part of the other one?
GAGs make proteoglycan.
All right, so the GAG is a part of the proteoglycan, so that's kind of the relationship between the two. That comes in handy for these questions about compare and contrast, and so you can say, well, this one is part of that one. The GAG is part of the proteoglycan. So this is the kind of stuff that's like inside. One of the main things you'll see about it is like your eyeball. It's got sort of jelly-like material in there, and there's various other places in the body where you kind of need a cushiony, soft kind of stuff, and that's kind of a cushiony, soft stuff. It absorbs water, so this whole thing is like a sponge and absorbs water, so it makes it sort of jelly-like material that can be useful in various spots. Okay, that's the same thing. And then finally, epithelial sheets. So epithelia is like skin, and not only skin, but other places that line sort of even in like some of the tubes inside your body, like you have like little tubes that lead down to your kidneys, and inside the tube there's like, you know, a space inside there. The cells that line inside of that, and they're touching that space, like there's air in front of them. They can also be epithelial cells. So if it's touching air, and again, a lot of times epithelial cells have a basal lamina. That's this yellow thing. It's collagen mostly, but some other stuff. And it's just a sheet of sort of non-living material that the cells are attached to. And so the cells will, you know, secrete this stuff, but then they kind of stick to it and have kind of a sheet. So that's the basal lamina, which often is kind of underneath a layer of cells, kind of supports them. It's sort of, you know, performs the same function as a cell wall, but it doesn't go all the way around the cells. And in a plant cell wall, every cell kind of makes its own wall, or here they're all kind of combining to kind of make this stuff underneath it. So the last main part we're going to talk about is all these junctions. So this is the sort of summary figure. You know, there's other figures that talk about each one of these, but this is the great summary figure. Like a lot of these chapters have great summary figures, so kind of memorize this stuff. These junctions. Tight junctions. I think we talked about tight junctions before, I don't know. But anyway, they're kind of sealing the gap between two cells. So this could, for example, be inside your intestine, like your small intestine, and on the top there is where the food's going through, and it's going to get absorbed by these cells, and then the cells are all stuck to this basal lamina at the bottom. And you don't want the liquid going through your intestine to be soaking down between the cells. So the tight junction is kind of sealing the gap, so there's no, it seals it tight, so there's nothing going between the cells. And that's kind of what it means. It's a tight seal between two cells. Of course, it's going to be three-dimensional. This only shows like a cross-section, so you know, the cell's three-dimensional. It has to go all the way around and kind of seal the junction between the cells. Adherens junctions and desmosomes. This is where different filaments from two different cells kind of connect. And the way you can memorize the difference is adherens junctions are where actin connects. So these are cross-sections of actin from two different cells. So I can get that pointer going on. Right here is actin. You see the cross-sections coming out of the board. So both cells have actin, and then they're kind of linked across between the cells, and that kind of holds the cells together, gives the whole sheet of cells some structure. Put this on, forget about this. And then desmosomes are basically the same thing, but here we're using intermediate filaments instead of actin, and they're still kind of joining two cells. And the way you can remember these, the adherens starts with an A, and so does actin. So adherens junction are actin connections, and then desmosomes are intermediate filaments. Gap junctions are where there's little holes between the cells, and like liquid can go back and forth between them. And hemidesmosomes are like desmosomes, but hemi means half, so it's like a half desmosome. So it's a cell connected to the basal lamina. So instead of two cells, that's a full desmosome, but it's only one cell instead of two, so it's only a half desmosome. And so it's connected to the bottom. And those are like intermediate filaments. So anything with a desmosome is going to have like intermediate filaments going on. So again, this is animal cells. Again, plant cells don't have intermediate filaments, although they do have microtubules and they do have actin, but they have cell walls, so they don't really need to have all these junctions like that. So this is more of an animal idea here. So we can look at a few of the tight pictures here. Again, here's tight junctions, and this is a better picture because it kind of shows like a more three-dimensional structure, and you see the junctions going down the cells, kind of sealing them up, so there's no space between for things to go between. And then here's some photographs. These are kind of cool. Here is either a desmosome or an adherens junction. Either way, you've got some kind of either an actin in an adherens junction or an intermediate filament in a desmosome. And then here's the two cells. And then here's how they're connected across the gap, and it's a cadherin. So cadherins work with both desmosomes and with adherens junctions. So the two cadherins, basically, one from each cell, and they come across and they stick to each other in the middle. So that's the cadherins. Here's another picture of them. This is a picture of actin filaments. So there's an adherens junction, and the cadherins, again, are the ones that actually cross between the two cells and hold them together. This is kind of cool, but we're going to kind of go through this. Here is desmosomes. keratin is one of those intermediate filaments. So here is keratin between two cells. Once again, you've got cadherins in between, and then the kind of connecting across the two cells. Beautiful picture on the left there. And yeah, it's the same thing again. Desmosomes, hemidesmosomes again. This one's got keratin again. Doesn't always have to be keratin. It's just some intermediate filament. Keratin, vimentin, some of those other intermediate filaments. And then here, these are integrins, because integrins, again, connect the cytoskeleton with the extracellular matrix. So now we're connecting not two cells, but extracellular matrix and a cell. So that's why those are integrin proteins, because they're connecting across the cell to an extracellular matrix and a cytoskeleton. Gap junctions. So these are like the little holes between cells. They're not just like random holes. There's these proteins that kind of line the holes and kind of form a pore, and they have to line up. So if they don't line up, it won't work. Obviously, the two cells have to line up their proteins to form a hole all the way through, and so cytoplasm can go all the way through. In some cases, for example, I believe like in your heart, where electrical signals have to go through all the cells, so they all beat at the same time, the signals can kind of go through these gap junctions, because it allows electrical current to kind of pass through. I'm trying to make out the picture there. It's hard to quite see it very well, but those are so gap junctions. And then this is the rare plant picture in this book, Plasmodesmata. How many people are familiar with this term, Plasmodesmata, or I think that's the plural. I'm trying to think what the singular is. This is the plant version of a gap junction. So in plants, of course, it's more difficult because you've got a wall in the way. So the plant version of a gap junction is the Plasmodesmata, or I'm trying to think of what the singular is. Plasmodesmosome? That doesn't sound right. And the cool thing about the plant version is the endoplasmic reticulum actually goes through the hole. So it's not just like water going through or cytoplasm. The ER actually goes through. Let's see if I can find the pointer to get it to work. Here, the ER is the yellow thing. It goes right through the hole as well. So you've got a wall here. So here's the membrane, and then the wall, and here's the wall, and there's a hole that goes all the way through, and the ER can actually extend between the two cells. So that's kind of cool, because then each cell doesn't have its own ER. They share the ER as well as the cytoplasm. And I think that's about the end of what we're trying to do as far as sort of extracellular matrix and stuff outside the cell. Is there anything else we want to talk about here? Desmosomes, hemidesmosomes, gap junctions. The book does talk a bit more in this chapter on stem cells. We should probably just mention, and this could be a whole nother topic, but we don't have time for it, but a bit on stem cells. What is a stem cell?
Non-differentiated cell.
Right, non-differentiated. So that means it's just sort of a boring round blob that hasn't yet formed into a muscle cell or a nerve cell or a kidney cell. When you form into a certain kind of cell, that's differentiation. And before that time, you're just sort of a cell. And the stem cell can divide, and when it divides, it makes another stem cell, and this cell can go on and differentiate. It may take a while for that to actually form into a new kind of cell, but that's what it does. And then the stem cell, as it divides, it always makes another stem cell, and then it makes another cell that can differentiate. So the stem cells will never run out because they always produce more of themselves. What kind of medical, why do we care about stem cells? I mean, sort of in a medical sense. Yeah.
Help you grow tissue.
All right, so you're going to grow new tissues, you need sort of stem cells. In embryos, when you're a little embryo, it's like when you're an 8-cell person, sort of all your cells are like stem cells. Every one of them could divide and become any kind of cell. At that point, and I used to have a better picture of this, but it came from another book, so I don't have it on me right now. But as you're an 8-cell baby, Each stem cell is called totipotent. I'm always getting the potent words mixed up. And that means it could divide and become anything. It has the potential to become anything. And then as you get older and older and the embryo gets a little more specialized, you still have some stem cells that cannot become just anything, but they can become a lot of things. And at that time, they are sort of pluripotent. And then as it divides and divides more and the embryo gets older and older, some of the stem cells are now more and more restricted in what they can become, and they can become a lot of things, but not as many things. And then they become multipotent. And I'm always getting these mixed up because they all sound like they're multiple and lots, but I'm pretty sure this is the order. This is the most able, these cells can become, the most kind of cells can become basically any cells, and these can become kind of a few kinds, and these kind of become sort of in the middle. Kind of how, as embryonic development proceeds, the cells get more and more kind of restricted in what they could become. Now the sort of medical idea is, can we regenerate tissues, using stem cells? So if you have like a spinal cord injury, can we get stem cells and they could become spinal cells? And then this is what Christopher Reeve, I don't know if you remember, he's the Superman in one of the 1970 movies, and he had a horse accident. He fell off a horse, or a horse fell off him or something. And he became paralyzed, and he was hoping for stem cell research. And that was going to allow stem cells to divide and produce new spinal cells, and they would put them in his spine. And that kind of stuff is now, it's not all the way there yet, but that's kind of stuff that people are working on and getting to work. This is why sometimes when someone has a baby, they save some of the cord, what was that cord called? The umbilical cord. Those cells have stem cells in them, and they could become anything. So you save those cells and freeze them, and if there's ever a issue, you can use those cells, and you can kind of guide them to become whatever other kind of cell, and they're going to match. Instead of using a donated cell from someone else that might not match, and it's incompatible, those will match. Some of the cells inside your bone marrow, they are sort of multipotent cells. They can produce different kinds of blood cells because they not produce just one kind of blood cell. They produce all kinds of blood cells, but only blood cells. So that's why they're multipotent and not totipotent. But bone marrow can produce all those as well. So there's bone marrow transplants going on so they can produce different kinds of cells in different individuals. The sort of ethical dilemma has come from embryonic cells from fetuses, aborted fetuses, because those are really young babies, and so that those cells could become all kinds of things. We have lots of medical uses for them, and there's research going on to those cells, and so the pro-abortion people are like, well, look, we can actually do research on these, and we're saving lives, and we're whatever, and the other people are like, no, this is only making abortion seem okay because you're doing something good with it, and that makes it more acceptable. Well, that's not good either. So that's kind of the controversy going on. The ultimate goal would be to take a skin cell, which is already very differentiated. It's not any kind of a stem cell, just a differentiated, but it has all the genes. Remember, every one of your cells has all your genes. And if you only knew how to treat it right, you could get that cell to become a stem cell and start dividing and producing anything in the body. It has all the right genes. All you have to do is make them turn on and off at the right times. And that research is actually, they've had some major breakthroughs in that. So you could take a skin cell and you could turn it into a stem cell just by treating with the right chemicals, and then you could just let that divide. You don't need embryos anymore. Your adult cells could do it all. They could divide and make any kind of cell you need. That if you had a spinal accident or any other kind of damage to any of your cells, you could take one of those cells and you could cause it to become any kind of cell that you needed and put it in. So that's kind of the idea of what's medically going on right now. And that's also brings up the idea of animal cloning, like Dolly the sheep. The whole idea of Dolly the sheep was, let's take say a skin cell from a sheep, and let's use that DNA to make another sheep. And right now, we don't have the technology to just take that cell and make that cell divide and produce a sheep. We have to still have an egg cell. So we take an egg cell from a lady sheep, which is called a ewe, and take out the nucleus. So now it's just an egg cell with no DNA, and then we'll put DNA from some other sheep's skin cell in there. And because now it's in an egg cell, it can actually divide and produce a whole sheep. And it only has the one parent because you took a skin cell, which is already diploid, had 2 of every chromosome, stick it into the egg, and that clones the sheep. that, again, is not completely easy, and there's issues going on, but there's more and more research. When they first tried this, it didn't work multiple times, and it never quite gave normal sheep. When I was in grad school, there was a individual in our program who was a grad student, and this person had their cat died, and the rumors were among the department that this person's cat was in the fridge or in the freezer, and he was just waiting to learn enough about biology that he could clone his dead cat, and that's why he was doing this. That's why he was in the program. But that's the idea of kind of cloning. And if you can, that gets to be scary. Dolly the sheep has been cloned. A bunch of other animals have now been cloned since that time. Humans not yet, as far as we know. I don't like that idea at all, of cloning humans. But there's all kinds of laws against it, but that's not going to stop somebody who really wants to. Yeah.
Can I ask why? Like, what do you see ethical reasons, or is it more
About humans, I just I haven't had any personal revelations on this subject, but I just feel like God wants everyone to have two parents and not one. And this way you can just take a skin cell and it's going to be a clone of you, and you're the only parent really. And I mean, yeah. But like I said, that's just my personal opinion on it. All right. Well, does anyone have any other issues on these? So for tomorrow's quiz, any one of these junctions might come up, as well as for Monday's test. So, get all these. I can't find the. Oh my gosh, it'll take me all day. Go back through all this and kind of memorize all these junctions and kind of what they are, as well as things like GAGs and a couple of proteins we talked about that are reported in the extracellular matrix, and proteoglycans. And I'll send out another email, and we'll do that on Friday, and then we'll start chapter 18 on the cell cycle on Friday, and then we'll continue that on Monday and continue that after break. Does anyone have or want to ask anything about anything on the test study? This is a shorter one. It's only got basically 2 chapters on this test for Monday, which is signal transduction stuff in 16 and cytoskeleton. Well, 3 chapters actually. The first part of 20 kind of mixed with 17.
I still couldn't find, I emailed you about it, but I still couldn't find like the 2B and the IIB, IIC.
Yeah, I wrote you back. So I believe on the class website under my notes. So this is like I've got my cytoskeleton notes. I kind of outlined from the chapter, and I use their big headings for like Roman numeral 1, Roman numeral 2, Roman numeral 3, and their smaller headings for like A, B, and C, and their smaller headings for 1, 2, 3, like this. And so, like 1A1 would be Roman numeral A, 1, and 1, or like Roman numeral 3, B, 1. And I've got one like this for chapter 16 as well. And I think it's on the class website.
So I can find the PowerPoint, but I couldn't find the.
Let me see here. Let me go look. All right, let's see here. Here's the PowerPoints, and the PowerPoints are actually the last version. They're down here somewhere.
Got it.
Yeah. All right. So down here, the notes down here, that's what these refer to.
Got it.
All right. Drawing. Things you should be able to draw. A sarcomere. So you've got the Z discs, and then you've got the actins that's coming in, and then the myosin that's overlapping. So if you can get something like a Z disc, and That's the actin, and then the myosin has the little heads, and they're kind of connected by these long, and that's the myosin. And each one of these has the same thing, all this myosin. So if you can get that, that's the sarcomere, and that's actin, myosin, and the Z discs, and of course there's a whole bunch of these together. You should be able to draw a cross-section of a microtubule. How many pairs, and then how many in the middle? Microtubule cross-section. So there's 9 pairs plus 2. And again, each pair is one complete circle and then kind of like that. So it's not total pair. It's like 1.5 microtubules, kind of. It's a weird formation. So there's 9 of these going around and then 2 in the middle. Anyone remember what the motor protein is that works on these to make them bend? Now, if you think carefully, we talked about 3 motor proteins. 2 of them were microtubule motor proteins, and 1 of them was an actin motor protein. So this should narrow down.
There's dyneins and
Kinesins. Yeah, and this is the dyneins that are moving on this one to make them move. Let's see. Troponin and tropomyosin. We didn't spend it. That was on the little movie we watched. I don't think we spent a huge. Let's see, that was in a different chapter. Basically, tropomyosin is like the protein that kind of coats on top of the myosin. So tropomyosin it basically blocks the myosin from being able to touch the actin. And then the troponin kind of pulls it off. So the troponin, tropomyosin, it's all kind of one big thing. Imagine that troponin is a little lever right here that kind of pulls up, and that pulls the tropomyosin off. And now the actin and myosin can touch. Calcium comes in, and calcium is what basically causes the troponin to change shape, pull the tropomyosin off, and then the actin and myosin can now touch, and they'll start to contract. So kind of review that little bit again. Go back and review the whole actin, that picture with all the actin in it, severing proteins, bundling proteins, cross-linking proteins, end capping proteins, sequestering proteins. I feel like there's some more, but just kind of memorize that list as well. We talked about differences between sort of actin myosin on one hand. Actin and microtubules on the one hand. Those are the fun ones, and then intermediate filaments on the other hand. So let's think about those, and then we'll go down to actin versus microtubules. So actin microtubules on one hand versus intermediate filaments. What are some differences?
Boring versus fun.
Okay, I knew someone was going to say that, so that's good, but let's get more specific.
Is that microtubules?
So microtubules and actin on kind of one category versus intermediate filaments on the other category.
Motors and one's more important.
Okay, one has motors and one doesn't. And by the way, why would you know that, even if you didn't know this, why would you know that intermediate filaments wouldn't probably have proteins? Motor proteins. I'll give you a hint. What's the difference between kinesins and dyneins? They go different directions. So think about different directions. Now think about intermediate filaments. They don't have different directions. They're symmetrical complete. So there's no plus end or minus end. So a motor protein wouldn't know which way to go because there's no directions. They're both exactly identical. So that's sort of one reason you wouldn't want to have a motor protein. So that's 2 things already. No motor proteins, and the intermediate filaments are completely, they don't have different kinds of ends. They're both exactly identical. And then what else makes them more boring?
Aren't they symmetrical, whereas the actin is not asymmetrical?
Yeah. What else? What are the two most exciting things that actin or microtubules do? Treadmilling and dynamic instability. So those are cool because they're always changing their shape. And intermediate filaments are just kind of static. They just kind of sit there and they don't have any changing of shape or size or anything like that. So, okay, now, and that's sort of the main things. Again, and we talked about intermediate. What's the word intermediate supposed to refer to? Size. So like the diameter. So the actins are the thinnest, and the microtubules are, of course, big round things that are hollow in the middle, are the thickest, and then the intermediates are kind of medium-sized on their diameter. So actin versus microtubules. What's kind of the distinction there? Okay, size is one of them. Obviously, the microtubules are tubules, are hollow inside, and the actins are not, and the actins are very thin. How many kinds of actin are there? Monomeric actin. How many kinds of tubulin have we talked about? I know there's at least that many kinds, but there might be more. All right, what kind of tubulin is there? Gamma tubulin, that's the little ring that can form the base where they can kind of nucleate from and start to grow from. And then what's the actual tube made out of? Alphas and betas. Yeah. And they're kind of, it goes alpha, beta, alpha, beta. So it comes in as a dimer, alpha, beta, dimer, and then as they stack up, it goes alpha, beta, alpha, beta, alpha, beta. So we got three kinds of tubulin already we talked about. How many kinds of actin have we talked about? I mean, actin is just one. It's just actin, and it's a sort of asymmetrical molecule, and that's why when you stack them up to make an actin filament, there's two different ends because they're not. It's like Legos. There's only one kind of Lego brick, but you stack them up. There's still two kinds of ends, but it's still just one kind of brick. So all the actins that I know of are just one kind of actin. So that's one difference. One similarity is they both have dynamic instability. There's a cap, and as that cap gets degraded, then the thing can fall apart. What's the difference in cap structure? Or it's not a very big difference, but
It's an ATP.
it's an ATP for actin, because it both start with an A, and then GTP for microtubules. And that's pretty much the difference. There's one kind of organelle in the cell in which microtubules can kind of be nucleated and they kind of stick out of it. And I don't think actin has a similar kind of little organelle. What's that organelle in especially animal cells? What was that? Okay, that's true too. I'm thinking about a place besides that, little round thing where the microtubules end up sticking out of and kind of growing from it, the centrosome. So the centrosome it's full of gamma tubulin where these can kind of come out from, and so the plus end sticks out and the minus end is inside here. And I don't think there's a similar kind of thing for actin. So those are some differences between microtubules and actin, even though they're more similar to each other than different in many ways. What processes are microtubules involved in? I can think of maybe three good things. Where do you find microtubules in a cell? All right, cell division, so the spindle pulling the chromosomes apart. What's another one of their functions where motor proteins are important? What was that? Okay, and what else do they do to organelles in the cell? So well, that's part of it. So again, they're like little tracks where the vesicles can move up and down, and then they help to kind of anchor in the ER, and the ER kind of attaches to it, and motor proteins help pull it out, so it's kind of spread out through the cell instead of all collapsed into one little spot. So you've got kind of a track kind of device, a spindle device, and of course the flagella. Actin has some similar roles as well, can be little tracks Of course, you'll find that in muscles. You find that especially in crawling cells where actin, as a cell crawls, it's actin filaments that kind of make up that leading edge. There's a nice figure in the book, and it's in a different chapter. But it's kind of the actin on that leading edge. It's kind of polymerizing to help the cell crawl forward. And then some of the cell cortex is actin, so kind of right under the cell membrane, kind of actin. And so we saw the actin connections between two cells that kind of has the same function as like an intermediate filament. It's going to connect through cadherins and kind of give support to the cells. So it has sort of multiple roles that way. So like kind of a support role, as well as a little microtubule, kind of same kind of idea of little tracks where things can go up and down, as well as sort of muscle and motors. And then intermediate filaments are pretty much just a structural kind of role. Let's see. Again, for the signaling stuff, those same pathways we talked about before, make sure you can draw those pathways leading to activation of protein kinase A, protein kinase C, calcium calmodulin, and MAP kinase. So go back and keep studying those. You know, maybe every morning just draw each pathway out once. That's your morning ritual. And if you can just do it every morning, by the time it comes to the test, you just kind of just do it again, and it's just. Make yourself a list of, if you haven't already, second messengers, and a list of enzymes that are kind of involved in those pathways. Just remember which things are the enzymes and which things are the second messengers. Gaps and gaps. All that stuff we've talked about. I feel like there's more that we could have on here, but. We're running out of time here. All right, I'm running out of questions and we're running out of time anyway. Anyone else have anything else they want to bring up while we're still here for the last minute? All right, in that case, I will send out another email once I figure out a quiz, and we'll do the quiz on Friday, and we'll start talking about cytoskeleton, and we'll do the test on Monday.